Hello students, today we are going to discuss about two important organelle of a eukaryotic cell, and that is lysosome and vacuoles. First of all, let me discuss about lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. Okay, this is the result of a Golgi apparatus. The isolated lysosomal vesicle, which is isolated from the Golgi apparatus, have been found to be very rich in almost all type of hydrolytic enzymes. So, hydrolytic enzyme, especially hydrolases of type, okay, that include lipases, proteases, carbohydrates, and you know very well the lipases are the responsible enzyme for the degrading, we can say, lipids. Okay, and proteases are the enzyme which are responsible for the breakdown of proteins, and carbohydrates are the enzymes which are responsible for breakdown of carbohydrates. Okay, so by this way, this organelle is rich with the hydrolytic enzymes, optimally active at the acidic pH, and mainly it is active on the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. Okay, so why this is very important organelle? Because you see here, this is the bacteria. Okay, or we can say any organism which is harmful to the cell. Okay, suppose it is entering inside the cell. Okay, so at that time when harmful bacteria enter inside the cell, what happened? Lysosome will play very important role. As I told you, this lysosome is rich with the hydrolytic enzymes, and any organism, okay, any living organism is made up of mainly four things, that is carbohydrates, protein, lipid and nucleic acid. So bacteria which enter will also made up of carbohydrates, protein, lipid and the nucleic acid. So when it enter, the enzyme which is present in the lysosome will degrade the organisms. Okay, so by this way, this organism will be killed out by the hydrolytic activity of the lysosome. Now, let me try to understand what is the formation of a lysosome, how lysosome will be formed. So, here you can see this is a rough endoplasmic reticulum. So, from the rough endoplasmic reticulum, some granules of a protein will be passed by using some transport vesicle. Okay, and the Cs region will receive it. Then, some process will occur inside the Golgi apparatus, and it will be received by the transphase of the, we can say this, this is the Golgi apparatus, and C phase, and this is the transphase. Okay, so by C phase it will be received from the endoplasmic reticulum, and to the transphase it will be reached. Okay, then these vesicle will convert into transport vesicle, and finally it will matures. Okay, it will be enlarged, and it will convert into lysosome. So by this way lysosome will be formed. So lysosome, as I told you, it is rich source of enzymes. So let me discuss which type of enzymes are particularly present in the lysosome. Essentially, the enzymes, about 30 to 40, are hydrolytic in nature. Okay, which is present in the lysosome. They can be grouped as follow. So some of them are a proteolytic enzyme, which include cathepsins. Okay, that is a proteinase, then collagenase, then elastase. Okay, then nucleic acid hydrolyzing enzyme, which include ribonuclease, which will degrade the RNAs, then deoxyribonuclease. Okay, then the third group is a lipid hydrolyzing enzyme, which will degrade the lipases, phospholipases, fatty acyl esterases. Okay, then carbohydrate splitting enzymes that include alpha glucosidase, then beta glycosidase. Okay, galactosidase, hyaluronidase. Okay, then aryl sulfatase. These all are the enzymes which are responsible for splitting the carbohydrates. Other enzymes will include acid phosphatase and catalase. These all are the list of the enzyme which is present inside the lysosome. Now let me try to understand how lysosome will work. So first of all, when a bacteria or any bacterium will be enter in the cell. Okay, and the process of a phagocytosis is initiated by the lysosome or a cell. Okay, see here, bacterium when received by the cell, okay, at that time it will be received in the form of this type of vesicle, and that is called phagosome. Okay, now this phagosome and this is the lysosome, both will merge together or fuse together, and it will be form a phagolysosome. Okay, phagosome and lysosome both will fuse together and form phagolysosome. Okay, and that's why here you can see it will digest all the particles present in the bacteria, and that's why bacteria will be killed out and soluble debris will be come out in the form of exocytosis. So by this way, whatever the harmful bacteria enter in the cell will be killed out by the lysosomal activity. Not only this lysosome will be degraded out or bacteria will be degraded out, whole cell may also blast and killed out. Okay, and that's why these type of lysosomal activity or lysosome itself will consider as a suicidal bed, because by the activity of a lysosome, whole cell may be blast and killed out. Now let me go through another organelle, and that is vacuole, and that is also a part of a endomembrane system. Okay, so the vacuole is the membrane bound space found in the cytoplasm. Okay, it contains water, sap, extracellular products and other material not useful to the cell. Means whatever the waste products are there will be accumulated inside the vacuole. And you know very well the large portion will be covered of the plant cell. You can see this is suppose plant cell. The large portion of a cell will be covered by the vacuole. Almost 70 to 90% of the cell portion will be covered by the vacuole. Okay, in plant cell the vacuole can occupy up to 90% of the volume of the cell. So this will be having a huge sized vacuole found in the. So this is the structure of a vacuole. See here, this is the vacuole structure. Okay, inside which there is a cell sap. Okay, then central vacuole is there. Then surrounding that, here you can see surrounding that one membrane is there, and that membrane is called tonoplast. So this is very important structure, that is a cell sap. Okay, then central vacuole and tonoplast. Now let me discuss some functions of the vacuole. First function, to isolating material that might be harmful or a threat to the cell. Okay, it will isolate all the material which are harmful to the cell. Then it will containing waste products, okay, small molecules and water. Then maintaining internal tug pressure, okay, within the cell, which can help to shape buoyancy, the force that make an object rise and move upward. So buoyancy will be maintained for the plant cell. Maintaining acidic pH, that is internal pH, exporting unwanted substances from the cell, okay, and allow plant to support structures such as leaves and flowers due to the pressure of the central vacuole. Okay, so by this way it will be allow the support of the plant structures. In seed, stored protein and needed for the germination are kept in the protein bodies. Okay, and which are modified vacuoles. So protein body is one type of a modified vacuoles. In a plant, the tonoplast facilitate the transport of a number of ions and even the material against concentration gradient. Okay, and that's why here you can see so many concentration of other, means their concentration is significantly higher in the vacuole than in the cytoplasm because of the transport against the concentration. Vacuoles play very important role in the amoeba. Let me see, in amoeba the contractile vacuole by using which they can engulf the food, okay, is important for the excretion also, okay, and taking the food also. In many cells as a protease, food vacuole are formed by engulfing the food particles. Here you can see it will participate in the engulfing the food as a food vacuoles. This is your homework for today's lecture. So you have to write down this homework in your notebook and submit it. And before we finish this lecture, let me go through an animation video. So look at this video.
Lysosomes, another member of the endomembrane system, are single-membrane vesicular structures formed during the packaging process in the Golgi apparatus. They contain hydrolytic enzymes such as lipases, proteases, and carbohydrases that digest lipids, proteins, and carbohydrates, and also nucleic acids. Apart from lysosomes, the endomembrane system also comprises vacuoles. Enveloped in a membrane called tonoplast, a vacuole contains water, sap, excretory products, and other materials not useful for the cell. The tonoplast of vacuoles in plant cells aids in the transportation of a number of ions and other materials against their concentration gradients into the vacuole, which is one of the reasons for the higher concentration of ions in the vacuole. In plant cells, vacuoles can make up 90% of the volume of the cell. These vacuoles also perform different functions. For instance, in amoeba, the contractile vacuole helps the organism excrete waste products and also in osmoregulation. On the other hand, in protists, food vacuoles formed by engulfing food particles provide nutrition to the organism. All the organelles of the endomembrane system work in a coordinated manner and play a vital role in biosynthesis, processing, packaging, transport, and secretion of proteins and lipids, as well as in storage and digestion of wastes.
In next lecture, we will learn very important organelle that is a powerhouse of your body, okay, and that is called mitochondria. So till that, goodbye.